Hello, I'm Mark Hughes. I'm a researcher at the Royal Botanic Garden, Edinburgh, and I'd like to give you some insight from the Edinburgh Begonia lab into the work we're doing into the world's fastest growing genus. I'll start with the acknowledgements. Working on such a massive genus is always going to be a collaborative effort, and these are some of the people I've had the great pleasure of working with over the years, and my co-authors, collaborators, and funders. It's been a great pleasure working with such a great group of people. So we work on Begonia because its distribution matches angiosperm richness hotspots. So you can see that it's really rich down the Andes, from the coastal forests of Brazil, and across the mountains of Southeast Asia. So nearly 2,000 species is an ideal model to explore the drivers of tropical species diversity. So we say fastest growing genus, not in an evolutionary sense, but in a taxonomic sense. And you can see that in the past 10 or 20 years, there's been a massive resurgence in interest in begonia taxonomy. And on average, about 60 new species per year are described. We expect to hit the 2,000 species barrier this year, so 2020 will be B2K, and this has been driven by largely through discoveries in Southeast Asia, not just by the Edinburgh lab, but by some teams in Malaysia and Indonesia as well. A lot of our discoveries come from our fieldwork program, like in this speculative expedition to Kalimantan, where we had no begonia records from whatsoever, but yet we found many new records and new species. There is just so much undiscovered and undescribed diversity. So we keep track of all this diversity in what we grandly call our Begonia Resource Centre, which is a database containing protologs, field photos, specimens, including all types, and distribution information, and all nomenclature and current synonymy. So if there are discoveries to be made on Borneo, there are certainly discoveries to be made on New Guinea, and this is work that's being led by Hannah Wilson for her PhD. And Hannah's interested in investigating this massive radiation of begonia on New Guinea. First of all, it's an inventory job. How many species are there and where are they distributed? And she's using herbarium phylogenomics, using hybrid bait capture, to build a time-calibrated phylogeny. And using this phylogeny, she's going to look for correlates of the speciation in terms of events like mountain building and also underlying genetic factors. Our initial look at the diversification rate, based on quite a small sample, and we compared it to diversification rates across sort of really rich floras like the Cape region and the Hawaiian archipelago, just for general dipole rates. And we can see that if we have got an estimate correct for 400 species in New Guinea of begonia, then it's going to be one of the highest angiosperm diversification rates that has ever been recorded. Our hybrid bait capture is looking really promising. We've got really good taxon and gene coverage, even from some fairly old herbarium material. So now the stage is set for us to get the phylogeny produced and understand this incredible radiation in its geographic and genetic context. So in complete contrast to New Guinea are the mountains of the Western Ghats in India, which are nowhere near as species rich, but much, much older. And this is the subject of Madhavi's PhD, and she's interested in the origin in terms of time and geography of the entire flora of the Ghats using begonia as a model and seeing whether the Ghats follow a museum or cradle hypothesis, i.e. to see if there's a signal of a relict flora in the Ghats. And this project really at the start needs to figure out the Gordian knot of Indian taxonomy and to get the names and the diversity sorted straight off. For instance, there are at least 14 sheets for this name as potential types, and Madhavi is doing genome skims and assembling organelle phylogenies for these to figure out the best way to typify and to understand the diversity before we go ahead and look at the geographic origins of the flora. So you might think that such a big genus would be the result of an adaptive radiation, but we can tell you that it's not. This is work done by Peter Moonlight, who found that Begonia niche evolution more corresponds to a random walk rather than to heavy selection pressure. So he looked at the evolution of the climatic niche of a couple of clades, one in section Goialdia and one from the Andes, and found that it matches a Brownian motion model. So where speciation events are not really linked to a big switch in niche. So if it's not an adaptive radiation, then what is it? So if begonia is not an adaptive radiation, what's driving it? So we've been investigating the evolution of the genome in begonia to get some more insights. We found that there's a genome duplication really early in the history of the Begoniaceae. And this has led to expansion of gene families. These larger gene families have more sequence variation, and some of these copies are potentially being repurposed. And we found that even between very closely related species pairs, like Begonia plebeja and Begonia conchifolia, we get different expression patterns of these copies. So Cynthia Fan is looking at this in more detail, focusing in on the genes that control leaf shape. So first of all, she's developing metrics to describe leaf shape variation in begonia and look at this in a phylogenetic context. She's then going to look at patterns in variation in key developmental genes, and then ultimately she wants to link the phenotypic variation back to the genetic map and do a QTL analysis to see how these leaf-shaped traits are linked back to the map and look for candidate genes. So Cynthia's done some really good work on scoring the phenotypes of our mapping population, which is between conchifolia and plebeja. First of all, using quite straightforward traits like circularity and perimeter, but also Fourier analysis as well. And she's got a really good morphological data set that shows a clear separation between the parents, the F1s, and the back cross. And this then sets the scene for us to be able to compare that to the genetic map. Cynthia is also looking at the evolution of key developmental genes in begonia, and the Kanadi genes control leaf symmetry, and obviously that's really important in begonia, which is defined by its asymmetric leaves. And she's found that in this gene family in begonia, we've got one loss and one duplication. We also see this expansion and change in copy number in the anthocyanin pathway. So there's a big variation in chalcone synthase copy number across species, which gives the potential for repurposing of these copies for different things. So what we've seen then is that the genomic evidence that we have, rather than pointing towards adaptive radiation, fits more with a model of mutation order speciation, which is where you can get a fairly similar phenotype, so without involving huge amounts of selection, but it's really due to these incompatible pathways underlying the phenotype that develop in isolation. And this can occur if you get low gene flow, you get high hybrid incompatibility, so you get similar fitness of these incompatible mutations, so again, lack of selection, and you get a period of allopatric divergence. So this is potentially perhaps a speciation model which could explain a lot of the diversity we see. But let's have a look to see if it will actually fit what other evidence we have for the genus. Well, it certainly fits what we see in terms of hybrid incompatibility. So if we cross things like Cardiacarpa and Euophylla, which have a fairly similar leaf shape, you get quite a sick F1 phenotype, and even more so with crossing with Nunfolia. So even with a similar leaf shape, there's obviously a really different genetic pathway behind that. So to go into this a bit deeper, this has been taken on by Luthia, who's looking at this genome dynamism aspect further. And she's looking at the variation in genome structure in terms of chromosomal changes and transposon activation, and seeing if this is correlated with high speciation rates. So she's been using a suite of 6 species picked across the genus and across the phylogeny to represent clades with different diversification rates. And she's examining the genome evolution, not at the level of the gene or gene family, but at the really big level in terms of genome size, transposable elements, and genome structure. So she's found, first of all, doing a survey of chromosome number, that we get a really big variation in chromosome number across the genus. So the phylogeny is there on the left, and in the Americas and in Asia, we get a massive diversification in genome in chromosome number, and this matches with the speciation rate. Africa's quite species poor, and therefore probably quite a low speciation rate. So this is the first insight that there could be some link there. And also Luthia has found a large amount of repetitive DNA in the Begonia genome, I mean massive amounts, and this again is linked with the clades that have the highest diversification rate. So this is further indication that a dynamic genome could be behind driving the diversification, the speciation we see in Begonia. Mutation order speciation requires a really high level of population isolation. And do we see this in Begonia? Well, yes, we absolutely do. So this was a survey I did as part of my PhD many years ago. I looked at 9 populations of Begonia sutherlandii, and I found that the population was overwhelmingly partitioned between populations, giving an FST of 0.9, which is the highest really observed in an angiosperm. So we get these massively isolated populations, and each one is starting to behave like an individual species. Are these tiny isolated populations of Begonia under threat from mutational meltdown under current climate change conditions if they exist in such isolated tiny populations? This is what Thibault is interested in for his PhD. Currently he's trying to develop a pipeline to look at historic material of Begonia species and modern day material, and assess if we can see a decline in heterozygosity and a decrease in genetic health of begonias over time. And he's using one of his model species is Begonia socotrana from Socotra, just south of the Arabian Peninsula. And we've got material from the late 19th century and very recently collected material, and we're going to see if there has been a decline in heterozygosity of this taxon as the region dries out. The evidence points to a non-adaptive, mutation order-driven diversification of Begonia, given the Brownian niche evolution, the highly isolated populations, and the dynamic genome evolution. So although it's not an adaptive radiation, it certainly is massive, and it certainly is very rapid. And we look forward to describing the 2,000th species this year. Thank you very much.